we're really happy you're here. My name is Mike Bagley, and I am fortunate to be the Vice President of Programs here at SoundWaters. And as part of that, I was also fortunate to spend a lot of time on boats with our SoundWaters research intensive scientists who you're going to meet in the next few minutes. And you'll hear more about their work this winter. Each of them spent a day a week with us on our research boat out in the Sound all winter, even the really cold days, until about, 3 weeks ago was the last time we went out. And they did some really interesting research, and the last couple of weeks, Tim, who you'll meet in a moment, has worked with them to put together the presentations you're going to see this afternoon. We're really grateful that you're here. The last time we got together in December was the first time we had a group inside our building for something like this, and this time that was when we were all masked, and this time there's a few less masks, and so each time we get together for SRI there's a little bit of happiness that comes with it. But we really appreciate you taking the time to come and see them. Without further ado, let me introduce you to Tim Abbott. Tim Abbott's one of our educators, and Tim has led the students the last couple of months in their work. And so Tim, we'll introduce you to them. Take it away.
Yeah. Hi everyone. So as Mike said, I'm Tim. I'm the SoundWaters Research Intensive Coordinator. And all of these students have chosen to spend cold days on the water with me instead of just going home and relaxing or doing their homework. They've all worked very hard both on the water, out in the elements, and also in our lab here, analyzing data, running experiments, and working with some pretty awesome science tools. And so without further ado, I'd like to welcome Lindsay Taylor from Stanford High, who's going to be presenting on periwinkle snails and their macroalgae consumption.
Snails and macroalgae consumption. So periwinkles are sea snail species that are invasive in the North Atlantic, and they actually came from Europe in the 1700s. They live on rocky coasts within the intertidal zone, which is an area between the low and high tide, and their diet consists of microalgae. And they're important because they maintain good algae levels, they break down plant material, which recycles nutrients back into the ecosystem. They are prey for many species such as sea stars and crabs, and they are indicated for marine health. So that's their diet, microalgae. There's also macroalgae. Starting with microalgae, the more common name is phytoplankton. They are unicellular, which means one cell. They are only visible under a microscope, and they float in all types of water, such as freshwater, saltwater, and brackish, which is a combination of both. And macroalgae, the more common name is seaweed. They're multicellular, visible to the naked eye, and they also are in water, but they're attached to rocks and shells. This is a picture of microalgae species under a microscope, and this is macroalgae species. The question is, are periwinkles only dependent on microalgae to survive? Can they eat macroalgae? And if they do eat macroalgae, would that have an impact on their growth or water quality? My hypothesis is that I think they will not be dependent on microalgae, that they will actually eat macroalgae. And the water quality will definitely be healthier because the periwinkles remove excessive algae, which maintains good levels of oxygen and chlorophyll. On to my experiment that I did. First thing I did was collect water from Holly Pond, which is actually right up there behind me, and I placed it in 5 tanks. And I collected a bunch of algae. The two I focused on was wireweed and rockweed. And if you look, you can see lime-colored algae, which is actually a species called mermaid tresses. On the first day, I weighed 10 grams of both algae, and I placed it in 4 tanks. And I also measured 16 periwinkles, and I placed 4 in 4 tanks. I kept the control just for the algae. And every week, I would take both the algae and the periwinkles out, dry it, and then weigh it. This is me in my eye. For me to measure the water quality, I use the sonde, and for it to work, you have to place the sonde in the water, and it will read many types of water qualities, but these are just the 2 I focused on. To make sure that the experiment was consistent, I kept track of the temperature and light using these loggers. And based on this graph, it kept track of the highs and lows of temperature and light. Looking at it, it stayed consistent most of the time, which means I had a consistent experiment. And to keep good oxygen levels, I used these bubblers in the tank, which you can somewhat see here. These were the results. These two tables show the weight of the wireweed and rockweed. Looking at both, you can definitely see there is a decline. Looking at rockweed, there was almost none left, which made the conclusion that periwinkles did indeed like the wireweed. They did not like the wireweed. And looking at April 6th on here and March 3rd on here, you will notice that there is a huge increase in tank 2's weight. And I believe this is due to a measurement error that I made while weighing it. And this is a picture of rockweed, and you'll notice that there are bite marks and holes within it. And the holes is because they eat with their radula, which is a bunch of many tiny teeth on like their tongue, and they scrape off the food off surface and drive it into their mouth. This is actually what it looks like. I think it's pretty disturbing. This is actually a time lapse of the periwinkles, even the rockweed. And remember, there was mermaid tresses on the rockweed as well. And notice that they eat it first. And notice how they do not eat the wireweed. And this is the wireweed, this is the rockweed.
Wow.
That's cool.
For the mermaid tresses, I actually did not measure it. I just marked it was present. And they clearly ate it because 3 out of the 4 tanks, it was gone by the 2nd week. And for the control, where there was no periwinkles, there was none left until April 6th. And this is the final weight of the periwinkles, and I found some interesting observations when I compared this to the algae weight. First off, looking at tank 1, they clearly ate the most. There was almost none left by April 6th, which explains why their weight declined because they had nothing left to eat. However, looking at tank 2, they ate the least, yet their weight increased, which didn't feel like right compared to tank 1. Looking at the control, they had the least amount of algae, but they had no periwinkles in the tank. Then I realized that it's because there was no nitrates in the tank that will help them grow, and nitrates come from periwinkle waste. And I noticed they had a linear decline for the control of the amount of rockweed. For the other tanks, it was an exponential decline, which I never made a conclusion as to why. And I noticed on the very last week, on April 13th, that all of the periwinkles' weight came to almost the exact same. There was no conclusion I made as to why either. For oxygen levels, it was the same percentage, about 100%, which is really good, despite having different levels of algae. But for the chlorophyll, I had the question in mind as, if there was an algal bloom, which is an excessive amount of algae, which can be very toxic to organisms, would periwinkles be able to maintain it? I said yes because the control had the most chlorophyll, which makes sense because there's no periwinkles present. Looking at tank 4, they had a huge decline in the amount, despite having the most, which shows that the periwinkles can be very beneficial. And for a conclusion, quick analysis, periwinkles, they did survive on macroalgae, especially the rockweed. They prevent high levels of chlorophyll. The oxygen did not decrease, and there was an interesting correlation between the periwinkles and the algae. More improvements for future experiments. I would like to have more samples, longer testing time, and better measurement accuracy. And after the experiment, I had a few questions. Would the ecosystem collapse if there was a slight decrease in periwinkles despite being invasive in a high population? Would algal blooms be more common in the Northeast if periwinkles never existed or were brought from Europe? And most importantly, why do the periwinkles only eat the rockweed? Why and how is it different from wireweed? Thank you very much. I hope for future experiments that I can find more conclusions, and I hope to do more research as to the relationship between the algae and the periwinkles, and for more improvements for the Long-Island Sound.
Woo!
accounts for having so much. I would think they are beneficial, and I would only know if there was none left, what would happen. So that's actually a question that I had in mind as well. Any other questions? Yes.
How did you go about weighing? Can you tell us how you weighed?
Oh yes. I can actually go back to many slides, but I did have a scale, and I actually did individually weigh each. This is like the scale I use. I just dried it and quickly weighed it, and I just recorded what was on there. Anything else?
Awesome.
All right. Again, awesome job, Lindsay. That was fantastic. I'd like to bring up our next two presenters, Grant Dietz and Julianne Dower. Grant from King School and Julianne from Darien High School.
So, hello again. As Tim mentioned, my name is Grant Dietz.
I'm Julianne Dower.
And over the course of this SRI, we looked at spring zooplankton abundances in the Stamford Harbor. So, first, we're just going to get into our hypothesis a little bit. We looked at the correspondence between zooplankton in the spring and some other statistics. So our hypothesis was as temperatures of both the sound and the air increase, as well as the chlorophyll in the water increases, so will the abundance of zooplankton in the sound. And this we thought because it means that as these increase, so will the necessary requirements for zooplankton and their food sources to survive.
So for introduction and background, just a quick fun fact is that zooplankton are expected to reduce due to climate change about 11 to 6% within the next century. And it's important because this is just a picture of the food. There's phytoplankton, which is plant plankton, and there is zooplankton, which is animal plankton, which is what we're focused on. So if you get rid of zooplankton, you're going to also all these species will start to decline as well on the entire chart. And our three main focus points for this presentation were kinds of plankton, reasons for abundance of plankton, and differences in between sites.
So I'm going to just go into some of the methods and how we've been about this. First, we wanted to look at some of the statistics we found in the water, and to do that, we used a YSI Pro DSS sonde, which is this thing here, which, as mentioned in the previous project, you put in the water and it will give you some readings and important measurements. What we focused on here was chlorophyll and the water temperature, especially at the surface level of the water. After that, we took a plankton net, which is this here, and we put it in the water and did something called trawling, which is when we put it on the side of the boat and basically towed it along with the boat for a certain amount of distance over the course of 3 sites. These 3 sites, which were different in, site here is near a waste plant, this site here is near the water, and this site is closest to the sound itself. So we did that for the same amount of time in each 3 sites. After that, we took our samples in a 125 milliliter bottle, and using a 6x6 gridded hydrophobic petri dish, which is this little thing right here, we measured the amount of plankton in each water drop under a dissecting microscope, which is that over there. And what we did was count the amount of plankton and compare it to the levels of chlorophyll or the levels of temperature for each day.
And here is our first graph of site comparisons. We have site 6 have the most zooplankton that we found, and site 1 have the least amount. And site 6 is the one that is closest to the actual sound, and site 1, I believe, is the one closest to rivers.
Yep, so this is site 6, and then this is site 1 here, and then this is site 8.
So basically the x's are median or the average, and then these bars down here are all of our medians, and then the lines at the top, they're the maximum value that we've gotten for each site. So then we have our line graph. It's telling basically the same information except we have dates here. So here you can tell for the first few days we really didn't get that much plankton at all. And sort of all at the same time, even though obviously site 6 did it at a much quicker rate. You can see that they all sort of increased at the same time. And then we have our site 6 have basically 1,500 plankton one day, and then they all start to decline also at the end, starting with April, which was interesting. And we have one outlier. This outlier is the 1,500 plankton that we got from site 6. This box is the average of all of our sites together, and then this is our max value without the outlier.
So basically, we decided that for our next few graphs, we weren't going to use this outlier because it messed with our data, and really with the outlier, it didn't really make sense as to why we were getting some of the numbers. So we decided to take it out, and we got much better results with some of our graphs. So first, we compared the amount of plankton over a course of time with the amount of chlorophyll in the water. And we'll explain a bit more why this amount of chlorophyll is important later. But right now, what you need to know is that there was a higher level of correlation, which we found seeing how close it was to this line here, which is basically like the middle of what happened. We found a higher level of correlation with chlorophyll and the amount of plankton. And as you can see, what we found is as the amount of chlorophyll increased, the amount of plankton also increased, which is what we expected to happen. Next, I'm going to compare the levels of the surface temperature of the water in Celsius and the air temperature in Celsius. And this is the surface temperature in Celsius of the water, and really this is not what we expected at all. There was pretty much what we found, no relation between the surface temperature of water and plankton, and we think the reason maybe of that is we only looked at this for 5 weeks. That's the amount of period of time we had to look at it. So perhaps, and most likely, if we looked at it over a longer period of time, there would have been more correlation between the surface temperature of the water and the amount of plankton. But from what we have, compared to other things, there's almost zero correlation, which we found really interesting and at times a bit confusing. But again, it could just be timing, we hypothesized. With temperature of the air, in contrast, we did find a higher correlation between the temperature of the air in the which we got by looking at the 5 days prior to that day and averaged those together, since the temperature that day wouldn't affect the zooplankton as much. So we found a higher correlation between the temperature of the air and the zooplankton, not as high as chlorophyll, however, much higher of a level than temperature of the water, which again was not much at all. So we were kind of relieved by that in a way and hypothesizing that yes, if we looked at this longer, all three would have had even more correlations than we found in the course of these 5 weeks.
Yeah, so basically there's some discussion, some possible food sources for the zooplankton that we found. So chlorophyll is found in phytoplankton, plant plankton, and that is what zooplankton eat. So the site 6, it's possible that with more water from the sound, there was more phytoplankton in site 6 than on the other sites, and that may be a reason why there were more zooplankton on that site. And there's also currents that could possibly affect where those zooplankton are at any time. So that's also could be a reason for what we found.
Finally, we'd just like to talk about how we could possibly continue this project in the future. With 3 things really: more time, more technology, and more data. So with time, obviously, as I've mentioned in the past, if we had a longer period of time, we might have found more correlation between the differences of plankton and what we're looking at. So again, time is just more important. With more technology, with higher technology, we might have been able to find would have been able to use larger samples of plankton, which we might even have found even more correlation with, as well as being able to maybe even find better results with higher levels of technology, as there is still possibility for human error without that. And finally, we want to have a more multivariable analysis of plankton levels, which requires higher levels of math, and and basically what it's saying is that one variable alone might not be affecting the levels of zooplankton. However, if we put this variable with something else, such as chlorophyll and currents together, that might have more of an effect and correlation on the amount of zooplankton over this winter and spring. Thank you for listening to our presentation.
Did you find a decrease in chlorophyll when you saw the decrease in zooplankton in April?
So if I go back to it, come on. So here, we actually hit a sort of cold front. It wasn't by a large amount of temperature, but there was still that cold front, and that kind of decreased plankton, chlorophyll, everything. So we hypothesized that's why there was such a large decrease in plankton, because we had numbers that were more similar to the first couple weeks than the week right before it. So we hypothesized that that cold front might have affected everything.
What would be an example of the advanced technology that
course.
you might like to use?
So this is a manta trawl, and basically what it is it's a larger trawl that can be used to collect more and larger samples of data. We were hoping that with technology on the computer we can use fine, but with higher amounts of data and trawling for longer periods of time, we can collect more plankton and have more set and confidence in our numbers. So with this better built technology for finding plankton and collecting it and looking at it, we would be able to have better data. So this is just one example, and one of the main examples that we can think of.
Excellent.
Our next presenter is actually continuing a past project. It's Owen Wyman from Stanford High School. And he's presenting on the yearly bottom temperatures of 3 sites in Stanford Harbor. Here you go, Owen.
Hi, I'm Owen. It's a long name right now. This is the yearly bottom temperature of 3 sites in Stanford Harbor. It's a longitudinal study, which means that it is a continuation of a study that I did last SRI thing, so the fall. It's a longitudinal study. Does anyone know what that is? Nobody. Oh, haha. A longitudinal study is essentially, it's an observation of repeated variables over a long period of time. So that means that it's essentially an expanded upon experiment with a lot of extra data over a long period of time. It doesn't necessarily have to be long, because mine isn't that long, but it just needs to be a time longer than a normal experiment. So my initial experiment asked, compared to years prior, has there been a significant temperature increase at a depth of 2 meters or below? I was curious about climate change and if it had any direct effect on the temps below 2 meters in Stanford Harbor. And I concluded that temperature variation can more accurately be seen over a large time span, hence the longitudinal study. I will keep this old research question I did, and I'll try to answer that question with more data points. So that's me. The materials I used for this leg of my study was just the data sonde. I think that you guys are familiar with that from the other projects. It's essentially just a big data collection device. It has a lot of crazy stuff on it, but it mostly, or at least for my case, it just takes temperature. It's like a big fancy thermometer. And last time, because we didn't own a data sonde, we had to use a lot of manual devices. But now we just have this, so it makes my life a little bit easier. That's a picture of me using it. The methods I used were every Monday, the team and I would go out to 3 sites in the Stanford Harbor, and we would lower it to a depth of 2 meters or below, and we would record the temperature. Then we would record our results in papers exactly like this behind me. I covered most of it up because it is very complex and kind of boring. Most of my project was spent recording all of this data. That's longitude, latitude, that is like, you wrote it, and time, and people, and all this crazy stuff, and that's just the first page. So the first and last days of the month were used as trip 1 and 2 for the data that you will be seeing right here. Trip 1, trip 2, trip 1, trip 2. Every month has 2 data points, because if I had a lot of extra data points, it would be kind of confusing and hard for at least me to read, and I know you guys can read. This is site 1. This is West Branch. That's the first site that I went to. And this, there's a data point right there. These are the 4 data points that I collected for this month and that I put into this graph. This is what these data points look expanded upon. This is all the data for March, February, and April. This is a strong positive correlation, as you can see. Strong positive basically just means that it keeps going up and that the data points are tightly put together. Sorry. This is East Branch. This is site 8. That is the waste treatment center right there. And again, you got 4 data points. And again, strong positive correlation. This is our last one. This is site 6. This is the branch closest to the ocean. And again, 4 data points. And again, strong positive correlation. Now, these 3 things are box and whisker plots. Does anyone know how box and whisker plots work? Anyone taking statistics? Well, I am. A box and whisker plot is kind of it's a way to visualize data, or at least wide data points. These 2 points are the outliers of the data, or sorry, not the outliers. These data points are the highest bounds of the data, so like the very, very warmest and very, very coldest. As you can see, site 8 has the most variation. It's also the warmest up there with 10. Site 6 is coldest, and site 1 is kind of in the middle. From this data, can anyone think or tell me why data or SDA 6 is coldest?
Closest to the ocean.
That's what I found. Yeah, it is closest to the actual sound. Boom. So again, my main result was there is a direct correlation between seasonality and temperature. Essentially, all the data I found correlates exactly with winter and spring. There is a strong positive correlation because it's just getting warmer. Like it's warmer in April than it is in February. Site 6 is much colder than sites 1 and 8. And that site 8 is much warmer and has greater temp variability. Now I believe that the variability might have something to do with the fact that the waste treatment plant gets flooded a lot, and there's a lot of different water conditions happening over here as compared to like sites 1 and sites 6. My conclusions. Yearly, there's not typically noticeable temp changes. If you want to do a longitudinal study about temp changes in the ocean, you preferably want like upwards of 10 years to do that kind of data. My data is a start, but needs more time to grow in order to compare future and past data. As you can see here, I started with 2020. The blue dots are 2020. I would like to expand upon all this data and continue for the next years to actually see if I can see a change from 2020. Does anyone have any questions? What's up, Christine?
So why did you choose to, I might have missed it, but why did you choose to study temperature longitudinally?
That's a great question. I was so curious about my initial project that I thought it would be beneficial for my curiosity to expand upon it, and also because I think it's important data.
Why did you initially choose it?
I'm passionate about the sound, and that's what you don't want to hear, right? No, I am. I do like the sound. And because, not for nothing, climate change does scare me, and I think I would like to know if it does affect my body of water, and I think it would be important for other people to know if it affects their body of water too, because we all live around here. Anything else?
Did you experience a difference using an actual thermometer versus a digital thermometer with accuracy or ease or anything?
It was definitely the data points using the sonde. they were much more accurate, like there was less variability than last time. I didn't do this really, because this is all the data points I collected. Oh, sorry. These are all the data points I collected in spring. I didn't do that with my fall data because I knew that it would be hard to visualize because all the data was too varied. Does anyone else have any questions? Thank you.
Thank you, Owen. That was fantastic. Our next two presenters. Our next two presenters are Will Durfee from New Canaan High School and Caleb Jacobson from Norwalk High School, and they'll be presenting on spring Stanford Harvard benthic biodiversity research.
So yeah, we're focusing our study on our biodiversity research. I'm Will Durfee. So we initially started this project with not really any goal in mind, just kind of collecting data, but as we went on through this SRI project, we found that the most interesting data to both of us was that of which we were catching while trawling, different than what Grant has explained earlier. We began to see a correlation between the increase in temperature in the sound and the amount of animals that we were catching with each trawl, which is our hypothesis.
So trawling, it's pretty much just putting out a big net behind you. We scrape the bottom of the floor, or at least as close as we can get, in order to try to get as much diversity as we can. There are different types of trawls. Ours, like I said, we hit the bottom, while other trawls might have gone more in the middle of the water in order to catch different region of fish. So some might get fish, ours got a lot of shrimp, for example.
Just like every other group, we use the SON to collect our temperature data as well as salinity. This was very important as it is half of our research.
So for our measurements, the majority of our measurements all went under 10 centimeters. There were a couple of outliers. For example, there was a species of sponge called the dead man's finger. Those, we've gotten ones that hit up to 14 centimeters. A lot of those were always bigger. So if we were to take those out of the example, I would say about 95% of our samples were underneath 6 centimeters. There was also a lot of variation between what animals we caught. Say if we caught a fish compared to a shrimp. I think a shrimp's kind of smaller than a fish. So with our shrimp, a lot of them averaged around 2 centimeters, though we have reached ones that got as big as 4.7 centimeters and some as small as 0.9 centimeters. And to figure out how large each of our samples were, we used a program called ImageJ. It's a program which allowed us to calculate how many pixels are in between a centimeter. So for us, we went 1/8 of an inch and used that 1/8 of an inch to then figure out how large our line was, and then translated how many pixels in between a line to figure out how many centimeters for a more accurate representation of how large it was.
We had a lot of diversity in this round of the SRI with 17 different species, including fish, jellyfish, sponge, crab, and mainly shrimp. The most abundant was the 7 spine shrimp, as you can see, which made up, around like 45% of what we caught.
That compared to last fall, we got more of a diverse spread than last fall's sampling, but although we got more of a diverse spread. They also got more shrimp than we did. So we got 17 species, they got 11 different species. So we just wanted to show how, although it's 2 different species, the main proportion of what we caught, the shrimp, stayed roughly the same.
Keep going back. Why we caught so many shrimp is because normally in the warmer seasons you'll find them closer to the shoreline, but because most of our data was done in colder times of years, the shrimp migrate out to deeper waters where we caught them.
So the water temperature, we figured out that the water temperature, as it started to increase, we started to see more species come out. The top one shows, the top graph showed all of our samples, but as you can see there is a large outlier, so we wanted to also show a graph without the outlier. Both of them do show a positive trend line, showing that it, as water temperature does increase, so does the total amount of fish that we caught.
So here's a map of where we did our trawls. It was close to the sound, but still in Stanford Harbor. This is a map of the sediment on the bottom. I know it's kind of hard to see, but this is the area where we did most of our trawls, which left us doing all of our trawls over silt or very fine sand.
So for our conclusions, we did find that our hypothesis was correct, as the water's temperature did get warmer. We had gotten a lot more species and a lot more fish to get out in general. And the biodiversity of what we found definitely increased along with our population density.
So for the future, to make this, I guess, a valid research project, we would like to continue de-trawling through all 4 seasons to get a better correlation between temperature changes. The larger sample sizes will make the evidence more concrete, as well as recording new species that show up with the warmer weather.
And so over our 2 months, we did find one catch that we loved by far, easily the best. It was our rock. We caught a big rock. It was about 38 centimeters. So thank you. Any questions? Yes.
Yeah, did you find a species that surprised you at all, or were they all the species you expected?
I got one. We had found a lion's mane jellyfish. That a lion's mane jellyfish, though it didn't really surprise us with Christina touching with her bare hand, with it being extremely poisonous or venomous. You know, it's not the best, but we thought that was kind of
That's cool.
stuck out.
We also found a couple of spider crab, which was very interesting.
So over a 2-month period, what was the temperature change that you pretty much?
It changed about 2 degrees Celsius.
Okay.
We started around
3.5.
yeah, 3.5, and it went up to around 5.5 towards the end. Yeah.
All right, so that doesn't sound like much. So it sounds like, yeah, you gotta go more months.
No, yeah. We want to, in the future, if we did continue this, we did want to figure out once it gets warmer, because this winter was kind of weird in a way. It was very cold throughout the entire time, even as spring started to come. So the temperature didn't change too much, but even that small degree of temperature showed so much more biodiversity. It would be cool to continue. Yes.
May have missed it, but what type of fish did you actually catch?
So the shrimp that we caught, or the fish that we caught, we found oyster toadfish, which I do not think we had a picture up on.
We have a picture of the tautog.
Yeah, and then found another fish called a tautog, which we had put here. Those are the 2 types of fish that we caught. We mostly caught shrimp and crabs for the most part. Anything else?
So when scientists talk about climate change in a couple degrees Celsius, and it's always a little unclear what that might mean, you guys just saw a couple degrees Celsius change. Do you have any thoughts about that and how you think about data globally?
I mean, I guess it's definitely alarming with temperature change and seeing this much change with just a couple degrees. And while this is a natural change over time, leaving the higher temperatures to get even higher than they ever were before definitely could lead to some consequences.
Along with also, like you said, that with how we saw so much more biodiversity as even at 2 degrees Celsius change, as temperatures do begin to rise and with global warming coming, we might be able to see even more species come out, but then that would also have to lead to the question, who's going to lead on top with like fighting over resources and food. Anything else?
How many oyster toadfish did you find?
We only found one. Yeah, we only found 2 fish. It was cool. Is that it?
Thank you, guys. They also started a nice little database with a ton of very precise measurements, which we're looking forward to continuing to use. So thank you guys for doing that. And our next presenter is going to be Mai Su, who is presenting on chlorophyll interactions at 3 different sites within Stanford Harvard. Take it away.
Okay, yes, that. Oh, you changed it back? Okay. Okay, so over here we have a picture of the data sound again. I won't go much into that because it was explained a couple times already, but yeah, Lindsay's holding that and she's holding it well. So to start off, we have some chlorophyll relations, but what is chlorophyll? So we have a picture of phytoplankton here. Chlorophyll is basically the measurement of all the plant life that's going on in the marine setting. So it can include like algae, the phytoplankton here. But with phytoplankton, chlorophyll allows plant material to do photosynthesis. So without photosynthesis, it can't use the sun's energy to basically just live. And plants are primary producers, so that means in the food chain, it starts with those. So herbivores come, carnivores come, and so on. So basically, it's just the building blocks of the food chain. Some aspects worth mentioning here are temperature. Low temperature seems to bring more nutrients, and also there's the process of cold water being denser. So when it's denser, it will get heavier, so drop to the bottom, and then this lighter water will come back up with the nutrients from the bottom, such as dead animal matter or just anything in general that will help them grow. And also from quick research, I think cold water can hold more oxygen, which is also good. We need oxygen, that's really good. So chlorophyll, just quantity. If you have too much or too little, that's going to be a problem. So too little, it seems a bit self-obvious. If it's too little, that means the plant activity doesn't have this component it needs to live, and then again, food chain. But if you have too much, you can have something called an algae bloom, and you could get something called an HAB, or harmful algae bloom. So with those, they can spread out across a water surface and completely block the sunlight from permeating between that level. So anything down here that is below this algae bloom, they will not be getting the sunlight. And a bunch of creatures and plants in general just need sunlight, so that isn't bad. And then finally on this page we have the rain. The rain, especially if it's heavy, can dislodge sediment from the ground, and it brings those nutrients in the runoff to the water sources nearby, and then they get some nice nourishment. Here we have a little slide of photosynthesis. So what's going in and what's going out? We have sunlight going to this plant here, carbon dioxide is going in, minerals are coming in here through the roots, and then water is coming in here. If this isn't the water, it would just be in the water. The minerals would be coming from the ground with the cold water dropping before, like I said. And then when they going up. So here you can conclude that with the more daylight you have, you get the temperature slightly increasing. Next, we have dissolved oxygen chlorophyll. In case you couldn't see it, because I made it really big. This is an MgL. This is not milligrams, which I almost put in here by accident. This is micrograms per liter. And this blue one here is the chlorophyll, so you can see it spiking up and spiking down. And then the dissolved oxygen, which is the red. And you can see here, all these edges here seem to directly correlate, even though it's slightly above it on the y-axis. So we can conclude from this that there's a direct correlation between the chlorophyll and dissolved oxygen. So when chlorophyll goes up, so does oxygen. When it goes down, so does the oxygen. Here we go. I have the sites that I regurgitated from my last presentation. Here's site 8. Again, this is the water treatment center, and it discharges a lot of fresh water. Site 1 is still experiencing some water movement here because fresh water is coming in from the Long River here, going down. Site 6 is where everything is just churning and swirling around. There's a lot of tidal action going along, and this is the most circulation out of the other 2 sites. Then there's the water and water kind of. Here we have the chlorophyll data by site. Site 1 is this blue one, site 8 is this orange one, and then site 6 is this gray one. A couple people did box and whisper plots again, so just briefly again, this is like the maximum amount here, this is the lowest amount here, and then this is like where most of the data lies, especially near that line in the middle. But we do have these little circles here. Now these are outliers. We've had a few gaps in the data and a few just numbers. I can assume that it's from the calibration of the data saw, and it was a little rocky on the start, and we also had to put a weight on it, so that measurements may have been a bit wonky from that. But again, this is micrograms per liter. Got up here. You can see that site 8 had most variability here with the maximum and the lower. It's like slightly higher, but not much, followed by site 1 and site 6. And then I think this last graph, we have surface chlorophyll and then surface temperature. So again, this is in Celsius and this is micrograms per liter. We can see that even though this is like a little bit spread out, a little hard to read, there is a trend line that is slowly going down. So we can see this in. Okay, so if you're kind of bored by now, you can just look at this guy. I don't mind. So with that, chlorophyll seems to prefer lower temperatures, so it will probably grow faster there since they have the nutrients before that are churning around. Site had the highest amount of chlorophyll. 1 was like in the middle, and then 6 had the lowest amount. And then we can see in the other graphs, direct correlation, chlorophyll with dissolved oxygen. And then the more daylight is the higher the surface temperature is. And this is like just, hold on, it's just like a bunch of stuff where I got my stuff. So if you want to ask me questions, I can do that. You guys can like drool over this, I guess.
I'm curious, I'm just confused a little. Does chlorophyll increase with temperature or decrease with temperature?
Chlorophyll increases the lower the temperature is.
All right, so it's inversely
It
proportional with
lets
temperature.
colder better because there's more nutrients there for it.
Okay.
But, so, is chlorophyll, what is chlorophyll? Is it the green, is it?
Chlorophyll is like the measure of plant life, so it can include phytoplankton, can include algae, and algae is like the green stuff. So that
The green stuff, yeah. Any idea why in cold water, you can see through to the bottom of it, it's crystal clear, the water is crystal clear, and then in higher temperatures, it gets kind of, obscure, muddy. Does that have anything to do with the chlorophyll liking?
Well, chlorophyll can be microscopic, so it doesn't have to be like visible to the eye. It's just like the measure of what's there. Even if it's like really, really, really tiny, just
but it likes cold.
From my research, it seems to like water. And when I looked online, and it seems to really thrive like Arctic, actually, so that can really show.
What time of the year will you see algae blooms the most? Isn't it like a pink
I don't know.
film on top of the water?
The chlorophyll A is actually green, I believe. And so the chlorophyll is like a light absorbing pigment that allows it to do the photosynthesis process. Algae, from what I've like physically seen, like my own experience, is I see it in the spring, but who knows?
It's in the hot weather, warm, very warm water can be a push to make the algae grow out of control.
Yeah, and then we can get chlorophyll algae blooms. Anything else?
Nice job.
Thank you, Mama. And our last two presenters of the evening are Skylar Flood from Greenwich High School and Allie Pinto from West Hill High School. And they're going to be presenting on marine debris on the main beach of Cove Island Park. Take it away. Thank you.
Hi, I'm Allie.
I'm Skylar.
And today we're going to talk about marine debris, the distribution of marine debris on the main beach of Cove Island Park. So let's begin by talking about why this is important. Most of marine debris we find in the ocean is plastic, about 80%. According to a recent study, there are about 5.25 trillion pieces of plastic debris in the ocean. This is extremely detrimental to the ecosystem because marine debris can injure or kill local wildlife, damage or degrade habitats, and threaten human health.
So our goals for our presentation were to find out what types of marine debris we found on the east shoreline of Cove Island, and what categories they fell into and how much there was.
So instead of an experiment, we did a research study, which is where we simply observed. And as you can see, it's an observational study. So in our case, the variable we were observing was the distribution of marine debris on the Long Island Sound. We did not compare anything, we just observed.
So we took a 50-meter stretch of shoreline and divided it into 10 sections, which we call transects. And each transect was 5 meters long. So we analyzed each transect one by one, and we did 4 each day of the trial. So here is the transects 1 through 10, and this is the east shoreline of Cove Island.
So after separating the transects, Skylar and I would pick out 4 at random. We spent about 10 minutes on each. We analyzed them separately, and we recorded the basic information such as the date, the coordinates, the transect number, and more, as you can see on the images to your right. So after we recorded all this basic information, we just collected any of the marine debris that was present in that given transect.
So we, once we had collected all of our debris, we marked off the debris by category on sheets and disposed of them properly, transect by transect. So examples of categories that we found were food wrappers, fishing lures and lines, and most frequently found hard plastic fragments.
These are images of some of the debris we found. Of course, we found a lot more. We found fishing lines, a lot of wrappers, and most of the debris we found was plastic.
So this is a pie chart representing all of the types of marine debris that we found and the quantity of each category. So hard plastic fragments made up of 59.4% of our findings, and the categories that were least frequently found were bags, fishing lures and lines, and food wrappers. So we found lots of hard plastic fragments. So the total number of
meters for the entire east shoreline is 261, and so we wanted to find the total average amount of debris for the whole shoreline. So this is just some boring math, but the average number of total debris rounds to 4. So it can be inferred that there are 4 total pieces per transect, and that divided by 5 is 52.2, which is the total amount of transects for the entire shoreline. So that would be 208.8, which is the final total average amount of debris for the entire shoreline on the east side of Cove Island.
So what did the data tell us? We learned that the majority of the debris we found were hard plastic fragments. This is very important because debris, plastic debris specifically, is very detrimental to local wildlife that live near or on the shores. For example, small fragments pose as a choking hazard for fish and seagulls. And
so other factors would be the beach cleanups that happen on Cove Island due to SoundWaters, which would definitely decrease the amount that we found on the beach, because there were some days that we found no debris. And another factor is the jetty on the far side, which would just impact the currents and what's washed up on the shore.
so there are some things that we could have done or that can be done in future experiments. For example, in future research studies, this leads us to think if we had collected the data over a few months rather than a few weeks, maybe we would have seen a relationship between the seasons and the distribution of marine debris. For example, would we have collected more marine debris in the summer instead of winter? These questions are very important because it leaves an open door for future researchers if they ever want to study these. But we can end on a positive note by focusing on what we can do. For example, limiting our use of single-use plastics and recycling can make a big difference in the long term. Also, we can organize and participate in beach and park cleanups regularly. Shout out to SoundWaters because they're always having cleanups. That's amazing. And lastly, we can encourage leaders to enforce policies and legislation. Thank you.
Could you tell whether this debris was making its way from the water or to the water?
It seems that it was coming, like, washed up on the shore from the water, but it's kind of hard to tell because we actually did find, like, a dog leash and, like, cigarettes, so it's kind of both.
What was the craziest thing you found?
We found like a total of like 50 cigarettes in one spot. Someone was definitely like camping out there, and like a jacket. But yeah, that was probably it. Yeah.
So you said a majority of the plastic or garbage that you were finding was hard plastic fragments.
Yeah.
Did you notice like a size commonality between them? Like were they really large pieces? Were they small, like teeny tiny?
Usually they were small, like this, about this size, small. Yeah.
Do you think that, how do those small fragments occur?
I believe perhaps through erosion over time because it seemed like most of the smaller plastic fragments have been washed up from the ocean from, I don't know, many other places perhaps. And I think it just broke down over time.
Awesome.
So you're impressed, aren't you? Yes. As you should be. So let's give all the presenters a Let's also congratulate Tim, who's lurking somewhere. Christina, who's lurking in the back. You can see the work that they did with these guys, and it's really impressive. I want to mention just a couple of things before we let you go. One is You heard the students talk about longitudinal studies, and one of the things we're most excited about with SRI, which we're going into our third summer of it this summer, is the ability to do what they're doing, which is collect data over a long period of time. So if you imagine next year and 2 years from now and 3 years from now, all the work that they've done will still be around, and the students that you'll be watching in 2 years will be comparing their data to what these students have collected. And that's something that's really exciting for us at SoundWaters. So they're kind of on the ground floor of that, but it's something that we'll be building over the years as we do this program throughout the year. The second is, you heard them talk about, a couple times reference the YSI data sonde and how important that was in their work. And you can see what using kind of modern tools, modern scientific tools did for them, just having that one. So we decided to build a building. Most of you, if not all of you, are aware of our Harbor Center on the other side of town. You can imagine what having a world-class wet lab like we're going to have over there will do for the work that future students are going to do, and some of these folks are going to do. So that's something we're also really excited about. Lastly, I just want to thank you again for coming. I know you have a lot of things, a lot of places you could be and a lot of things that you could do. It's really important for them to learn. One of the skills I'll learn as scientists is how to present data, and
Very good.
they can do that to an empty room, but it's much better when they do it with a lot of mean faces staring back at them. And you certainly fit that bill. And to reward you for coming, we have a real beautiful buffet in the back of the room. Please help yourself. Ask students any questions, and we will look forward to seeing you next time we do this. Will be next December, and it will be in the Harbor Center, which is what we're